In December 2023, the FDA approved Casgevy, the world's first drug based on CRISPR technology. The treatment addresses sickle cell disease. A microscopic glitch in the DNA causes red blood cells to collapse into a rigid crescent shape. These misshapen cells block blood vessels, causing patients to live with chronic, intense pain. Instead of just managing the pain, this drug rewrites the patient's genetic code. It edits the source of the malfunction to provide a functional cure. For these patients, a single sequence correction successfully stops a cycle of inherited pain that was once considered permanent. This technology originated as a bacterial immune system. Bacteria use CRISPR to identify and chop up the DNA of invading viruses to stop an infection. In 2012, researchers led by Jennifer Doudna and Emmanuel Charpentier successfully reprogrammed this defense mechanism into a set of programmable genetic scissors. The traditional version of this tool, using the Cas9 enzyme, works by creating a double-strand break. It cuts through both sides of the DNA double helix. When severed, the cell attempts repair using a pathway called non-homologous end joining, or NHEJ, to stitch the broken ends. This emergency process is frequently inaccurate, resulting in random deletions or insertions that can disable the gene entirely. While Cas9 is effective at disabling genes, severing the DNA is often too destructive for subtle medical corrections. To treat more complex diseases, researchers needed a way to edit the code without breaking the strand. Modern genetics utilizes a triage matrix. Because genetic mutations vary in their structure, scientists must match the specific error to a specific molecular tool. Traditional CRISPR-Cas9 is the heavy-duty option in this matrix. Its ability to disrupt DNA makes it the primary tool for specific tasks. It is used in applications where breaking a gene is the goal, such as disabling a malfunctioning gene or engineering immune cells to identify and attack cancer. Cas9 remains a standard for gene disruption and large deletions, but it is sidelined when the patient requires a more delicate sequence rewrite. In 2016, scientists introduced base editing. This was the first tool designed to chemically alter DNA without severing the double helix. Base editing uses a modified Cas9 that does not cut the DNA. Instead, it carries an enzyme that chemically converts one genetic letter into another, while the rest of the helix remains intact. This provides high efficiency for correcting single-letter mutations. This precision is the method currently being used in clinical trials for blood disorders like beta thalassemia. Base editing functions as a precision eraser for single-letter errors. It offers safety and efficiency for simple corrections, though it cannot write new, complex sequences. Base editors can only address 4 of the 12 possible DNA letter conversions. This leaves many complex genetic diseases unaddressed. Prime editing, developed in 2019, provides a search and replace function. It allows scientists to write brand new sequences directly into the genome. It uses a nickase to cut only one side of the DNA. Then, a reverse transcriptase writes new genetic information directly into the gap before the strand is sealed. Prime editing can address multi-letter mutations, such as those found in cystic fibrosis and Huntington's disease. It fills the gap between the power of Cas9 and the precision of base editing. This completed toolkit has broad implications. Scientists can now target 89% of known disease-causing variants listed in the ClinVar database. This precision is being applied in agriculture. Researchers are using these tools for the rapid creation of crop species that can survive in extreme climates. In public health, experiments are underway to edit the genomes of mosquito populations in order to stop the transmission of malaria. These advancements show that genome editing is becoming a fundamental technological infrastructure used to solve problems across the biological world. For decades, genetics was focused on reading and mapping the human genome. Modern science has moved toward active intervention. The specific type of mutation now dictates the tool used for the cure, whether they need the heavy scissors, the precision eraser, or the search and replace editor. With the ability to edit genetic code established, the focus has shifted to matching the right molecular tool to the right patient.